close your eyes and imagine the deadliest animal in the world. Do you see a shark or a snake? You should be picturing a mosquito. Mosquitoes transmit dozens of pathogens, infecting more than 700 million people, resulting in over 1 million deaths each year. In the US, West Nile virus is the most common mosquito-borne disease. The two main mosquito species in Baltimore and DC are the tiger mosquito and the northern house mosquito. Neither are native, and both are capable of transmitting West Nile. Unfortunately, residents in socioeconomically disadvantaged areas are at an unjust risk of West Nile exposure. Previous studies in Baltimore indicate that mosquitoes are more numerous and longer lived on lower income blocks, and by analyzing PCR data, I found that they are also more likely to be infected with West Nile. Why is that? We need to understand what factors create more dangerous mosquito populations so we can better control them. Larval mosquitoes in urban areas live in artificial container habitats where water can collect, like pieces of trash or flower pots. They feed on microbes supported by decaying organic matter, like leaves that fall from above into these containers and break down. The quantity and quality of those leaves can affect life history traits such as survival, development rate, and adult body size, all of which influence the likelihood of disease transmission. Previous studies have found that non-native plant leaves foster a higher rate of survival, faster development, and larger adults than native plant leaves do. Additionally, the plant resources available to these two common mosquito species determine whether they are able to coexist or whether one species outcompetes the other. The northern house mosquito plays a larger role in amplifying West Nile amongst birds, and the tiger mosquito may be more likely to bridge it to humans. So disease risk is greatest when these two coexist, and that's more likely when the plant resource base is non-native. I surveyed plant communities across socioeconomically diverse neighborhoods in Baltimore and DC, identifying which plant species were present in the canopy in order to understand what leaves would be available to larval mosquitoes. I found that the most common plant species on low-income blocks were non-native, whereas high-income blocks hosted mostly native species. I hypothesized that the non-native plants are playing a role in the proliferation and coexistence of these two non-native mosquito species on low-income blocks. The next step is to raise both mosquitoes in the lab on plant leaves that I found to be representative of socioeconomically diverse neighborhoods. While other studies have investigated the influence of plants on these mosquitoes, my research will be the first to do so along socioeconomic gradients in urban areas. The results from my work can give us insight for controlling mosquitoes by managing vegetation, hopefully addressing an environmental injustice for urban residents.